rough endoplasmic reticulum, smooth endoplasmic reticulum, and then cell trafficking. For the rough endoplasmic reticulum, is the site of synthesis of secretory exported proteins. So they are the proteins that are going to be exported outside of the cell, and also is the site of synthesis of unlinked oligosaccharide addition to lysosomal and other proteins. Actually, not the synthesis is just the size of the linkage or the addition of the unlinked oligosaccharides to the lysosomal and other proteins. We have the nestled bodies within the rough endoplasmic reticulum in the neurons. That's specific place. They do synthesize peptide neurotransmitters for secretion. So that's what creates the peptide neurotransmitters that are going to roam around the body. We have the free ribosomes. They're unattached, just like free. They're unattached to any membrane. They are the site of synthesis of cytosolic, peroxismal, and mitochondrial proteins. Cytosolic, peroxismal, and mitochondrial proteins. This is just for memorization. Unlinked glycosylation occurs in the endoplasmic reticulum. We also have the mucus secreting goblet cells of the small intestine. We know a lot of mucus production occurs in the intestines, and also an antibody secreting plasma cells are rich in rough endoplasmic reticulum. So the mucus coming out of from the goblet cells in the intestine, and also the plasma cells, they are which are secreting antibody secreting antibodies. Yeah, they're secreting antibodies. They're rich in the rough endoplasmic reticulum. As we said, it's for the proteins that are going to be exported outside of the cell. That's why proteins within organelles, just like endoplasmic reticulum, Golgi bodies, lysosomes, are formed in the rough endoplasmic reticulum. So the proteins as well that are within the organelles, like within the plasmic reticulum, or within the Golgi bodies, or within the lysosomes, they are also formed in the rough endoplasmic reticulum. So that's for rough endoplasmic reticulum. Let's see, we have something. So for the mucus, secreting goblet cells of the small intestine, the mucus release is increased in inflammatory states. Stool will have visible mucus in conditions such as inflammatory diarrhea, giardiasis, and cholera and Crohn's disease. Yeah, makes a lot of sense. And also protein what? Proteins within the organelles. It's along with integral membrane proteins of the nucleus and the cell membrane, and proteins within the endoplasmic reticulum, Golgi network, and lysosomes. So these are the proteins that are in these places, like the endoplasmic reticulum, Golgi network, and lysosomes, in addition to the integral membrane proteins of the nucleus and the cell membrane. For the free ribosomes, it's what is the role of free ribosomes? It's just as we said, it's secretion of proteins in cytosol, nucleosol, peroxisome, and mitochondrial proteins. We also have the mitochondrial proteins. The mitochondrial proteins. So what are the mitochondrial proteins? They're like the proteins within the mitochondria contain the nucleosol, peroxisome matrix, nuclear encoded mitochondrial proteins. Nucleosol, peroxisome matrix, and nuclear encoded mitochondrial proteins. You also have all ribosomes begin protein translation in the cytoplasm, but some translocate to the plasmic reticulum. So all the ribosomes start the process of protein translation within the cytoplasm, not the nucleus. But some of those ribosomes, once they start the protein translation, they translocate, they change their place to go into the rough endoplasmic reticulum. So we also have ribosomes attached to the endoplasmic reticulum via translocone. It's a protein complex containing ribophorines that bind large 60S subunits. Okay, so in case we do have some ribosomes that do attach to the endoplasmic reticulum, they are attached via a process or like some kind of protein that is called translocone. Okay, so there's protein um, between these two things. This protein complex contains ribophorines that bind the large 60S subunits, because we know that the ribosomes are contained, they do have two portions, the small one and the second one that is the big. So I guess this is the 60S protein, the big one that is connected to the ER, the plasma reticulum. For the smooth endoplasmic reticulum, it's the site of steroid synthesis. So, and detoxification of drugs and poisons. Smooth is the site of steroid synthesis and detoxification of drugs and poisons. It lacks surface ribosomes because it's smooth, so it doesn't have any ribosomes attached. Location, it's the location of glucose 6-phosphate, 6-phosphatase. That's the last step in both glycogenolysis and gluconeogenesis. So the last step of these two processes, the glycogenolysis and gluconeogenesis, is occurring in this smooth endoplasmic reticulum, and that is the location of glucose 6 phosphatase. Hepatocytes, steroid hormone producing cells of the adrenal cortex and gonads are rich in smooth endoplasmic reticulum. So the hepatocytes plus the cells of the adrenal cortex and gonads, which are responsible for secreting steroid hormone, they are rich in with the smooth endoplasmic reticulum. It also plays, so this smooth endoplasmic reticulum also plays a role in lipid synthesis and carbohydrate metabolism. Lipid synthesis and carbohydrate metabolism, plus smooth endoplasmic reticulum, it has the role, it's involved in synthesis and processing of hydrophobic compounds, including lipids, phospholipids, cholesterol. So hydrophobic compounds like lipids, phospholipids, cholesterol, just like the steroids. Plus, cholesterol is initially converted to pregnenolone in the mitochondria and then transferred to the smooth endoplasmic reticulum. So cholesterol initially was converted to pregnenolone in the mitochondria and then transferred to the smooth endoplasmic reticulum. Okay. Yeah, we talked about that. So for the self-trafficking, it says that Golgi is distribution center for proteins and lipids from the endoplasmic reticulum to vesicles in plasma membrane. So that's the factory, and this is the I don't know, distribution point. That's the Golgi apparatus. So the Golgi apparatus is the one that's responsible for distributing those proteins and lipids from the endoplasmic reticulum, whether it was smooth or whether it was rough, into like going to the vesicles and plasma membrane. Post-translational events in Golgi include, okay, so that's post-translational, uh, basically after the protein has been created. They do occur in Golgi apparatus. They include modifying N oligosaccharides on um, asparagine, adding O oligosaccharides to serine and threonine, and adding mannose 6-phosphate to proteins for lysosomal and other proteins. So basically, a few steps over here, a few functions. First is like modifying the N oligosaccharides on asparagine specifically, in addition to second adding O oligosaccharides to serine and threonine, plus three, that is the addition of mannose 6-phosphate to proteins. And the reason for that to for in order to be used for lysosomal and other proteins. Okay, so endosomes are sorting center. That's for Golgi apparatus. Okay, now we have another apparatus, that's the endosomes. So the endosomes, they're the sorting center for material from outside the cell or from the Golgi, sending it to lysosomes for destruction or back to the membrane or Golgi for further use. So in order to decide where is this material going to next, we have the endosomes. They decide if this material that is coming from outside of the cell or the material that came from the Golgi apparatus, should it be sent to lysosome for destruction or should it go back to the membrane or to the Golgi apparatus for further use, just to reuse it. Um, we have the one cell disease. It's inclusion inclusion cell disease, mucolipidosis type 2. So there are few names. Uh, one cell disease, inclusion, also called inclusion cell disease. Basically, this is an I, not just one. So this is inclusion cell disease, or it could be called mucolipidosis type 2. It's an inherited lysosomal storage disorder, a problem with the lysosome storing function. It's an autosomal recessive. There is a defect in the N-acetylglucosamine 1 phosphotransferase. Wow. There is a failure of the Golgi to phosphorylate mannose residues. So the problem here is a defect with the Golgi's function, and that function is phosphorylating the mannose residues. This is what the Golgi should have done, but the Golgi cannot do it, so that's why we have a problem. Um, Basically, the phosphorylation of mannose residues should reduce the mannose 6-phosphate, and that is on glycoproteins. When we do have a defect with that phosphorylation of mannose residues, we have less mannose 6-phosphate on glycoproteins. That leads to enzymes secreted extracellularly rather than delivered to lysosomes. So lysosomes deficient in digestive enzymes. So basically, because of this defect, the enzymes are going to be secreted extracellularly, like outside of the cell, but they actually should have gone to the lysosome, should have been contained within the lysosomes. And now we don't have enzymes within the lysosomes because they went outside the cell. So the lysosomes are being deficient now in digestive enzymes, which leads to buildup of cellular debris because the lysosomes cannot like degrade that. That's why we have inclusion bodies within the lysosomes. That's why it's called inclusion cell disease. It results in coarse facial features, gingival hyperplasia, corneal clouding, restricted joint movements, claw hand deformities, kyphoscoliosis, and increased plasma level of lysosomal enzymes. Increased plasma, of course, because it's being these lysosomal enzymes that should have been contained within the lysosomes are being secreted outside the cell. That's why when we check the blood, we see their level increased in plasma. The symptoms are similar to, but more severe than Hurler syndrome. So it's kind of look like Hurler syndrome, but it's even more severe. It could be fatal in children. So often fatal in children. That's inclusion cell disease. And that was the defect with the N-acetylglucosamine 1 phosphotransferase, and that's an enzyme. So we have a defective enzyme that causes failure of the Golgi to phosphorylate mannose residues on glycoproteins. Let's see what we have here. So for the clinical features of inclusion cell disease, it's a fatal autosomal recessive lysosomal starch disease due to failure of phosphorylation of mannose residues. Proteins cannot transfer to the lysosomes, accumulation of cellular debris in the lysosomes because it cannot be degraded. There is inclusion cell disease. The clinical picture includes coarse facies, corneal clouding, restricted joint mobility. Okay. We also have some other disease. So it's not a disease, it's a particle. It's signal recognition particle. Signal recognition particle, it's abundant cytosolic ribonucleoproteins that traffics polypeptide ribosome complex from the cytosol to the rough endoplasmic reticulum. So it's some kind of uh, ribonucleoprotein that should traffic polypeptide ribosome complexes. So, by the addition of polypeptides to ribosomes, they form complexes. They're called polypeptide ribosome complexes, and these complexes should be trafficked from the cytosol to the rough endoplasmic reticulum. And the particle that should do that is the cytosolic ribonucleoproteins. Absent or dysfunctional signal recognition particles leads to accumulation of proteins within the cytosol, like they're not being trafficked to the rough endoplasmic reticulum. That's why they stay in the cytosol. That's for signal recognition particles. We also have vesicular trafficking proteins. Okay. Okay. For the signal recognition particle, we have which of the following best explains why proteins synthesized in the rough endoplasmic reticulum and secreted from the eukaryotic cells have a shorter length than would be predicted by their respective messenger RNAs is due to that leads to that's because of cleavage of 5' segment of nascent polypeptides being synthesized on polyribosomes in the endoplasmic reticulum and not by non-specific protease activity during passage of these proteins through the endoplasmic reticulum membrane. So the question was why these proteins are being synthesized in the endoplasmic reticulum, like and secreted from the eukaryotic cells. They do have like shorter length than what is supposed to be when we compare it to the messenger RNAs. And the reason for that is because we are doing some like cleavage of one of their ends, one of those ends for those proteins. That why they look shorter than we would expect when we compare them to their messenger RNA. Okay, and that cleavage has occurred in the 5' end. The explanation for that also is, and the reason for that is like it happened in the endoplasmic reticulum, not by a non-specific protease activity during their passage uh, from the endoplasmic reticulum to the endoplasmic reticulum membrane. Hmm. Okay. So it's basically like a specific process of cleaving that part, that end of the protein, and it's not happening because it's just passing from one place to another place. We do have a protease over here, so it's not due to this protease. The explanation is that the protein synthesis starts on free ribosomes in the cytoplasm and continues in the endoplasmic reticulum. So the protein synthesis, it happens in the free ribosomes. We already talked about that, right? Free ribosomes. There you go. That's the free ribosomes that we talked about. They're not attached to any membrane, and they are the site of synthesis for cytosolic, peroxisomal, and mitochondrial proteins. So protein synthesis starts here in the free ribosomes in the cytoplasm and then continues to the endoplasmic reticulum. Okay, almost all proteins that are transported to the endoplasmic reticulum have a sequence consisting of 5 to 10 hydrophobic amino acids on the N terminus. All of those, like basically almost all of them, that are being transported from the free ribosomes to the endoplasmic reticulum, they do have a sequence of nucleotides that's 5 to 10 hydrophobic amino acids. Like amino acids, they are hydrophobic, and within a specific sequence, they do exist on the N terminus. Okay, so the proteins is guided to the endoplasmic reticulum by signal recognition particles. As we said, it's the particle that should move these proteins from their uh, like synthesis site to their destination, and that is the endoplasmic reticulum. So these protein is guided to the endoplasmic reticulum by a signal recognition particle which moves between the endoplasmic reticulum and the cytoplasm. So these particles, they do move back and forth. The N terminus, one end of polypeptide chain, that is the protein. Remember the protein that we're trying to transfer from one place to another, it does have an N terminus, and that is one of its ends. This end um, contains a few amino acids. Like over here, we do have some amino acids that they work as like a tag, as an address tag, which are removed when the polypeptide reaches the destination. So whenever this protein, so these amino acids, these tags are going to be removed once this protein reaches its destination. And that's it. The N terminus corresponds to the 5' end, as the carboxy terminus is for the. So since we know that the protein has two ends, of course, one is the 5' end and the one is the 3' end. The 5' end is the one that has the N terminus, while the 3' end is the one that has carboxy terminus. Okay. Um, Now for vesicular trafficking proteins, we have multiple names. There is COP1, COP2. COP1 is the Golgi, then leads to Golgi retrograde, then cis Golgi, then endoplasmic reticulum. So it's vesicular trafficking proteins, which is supposed to mean that they're the proteins that traffic the like traffic. Oh, these are the components that do the trafficking in the in the vessels, not vessels, like vesicles. Yes, in the vesicles, there's the different vesicles. Okay, so we have COP1, we have COP2. COP1 is leading to trafficking from the Golgi to the Golgi itself. That's why it's retrograde. We also have cis Golgi that causes trafficking to the endoplasmic reticulum, so like from cis Golgi to the endoplasmic reticulum. So that's COP1. For COP2, it causes the trafficking from the endoplasmic reticulum to the cis Golgi. That's anterograde, okay? COP2 from endoplasmic reticulum to the cis Golgi. That's the anterograde. Two COP step forwards, that's anterograde, and the one is step back retrograde. Okay, that's for just memorization. We have clathrine. Clathrine. That's trans Golgi, okay? Lysosomes. Uh, so the clathrine leads to trafficking from Golgi to the lysosomes. That's one. And then we have some different thing that is from plasma membranes to the endosomes. Okay. Then clathrine has two functions. One is moving from trans Golgi to the lysosomes, and the second is from plasma membrane to the endosomes. That's first, that's second, okay? So clathrine trans Golgi to lysosomes and plasma membrane to endosomes. Receptor-mediated endocytosis. This is where the receptor-mediated endocytosis occurs because of course, the pathogen or whatever thing that is going to enter the cell is first going to encounter the plasma membrane, okay? And the plasma membrane is going to be like from that material is going to be transported from the plasma membrane to the endosomes. That happens by the clathrine. The clathrine uh, mediates this process. That's why it's called receptor-mediated endocytosis, like from outside the cell to the plasma membrane to the clathrine to the endosomes, and then we end up with the process that is called receptor-mediated endocytosis, just like LDL receptor activity, low-density lipoprotein activity. Now, let's take a look over here. Okay, so basically we start with the plasma membrane. And the plasma membrane is going to encounter whatever that's outside over here, and then takes it in in a process called like to the early endosome, and within the early endosome, it ends up being in the late endosome. So it makes sense. That's one first, that's second, and then from the late endosome, it either goes to the Golgi apparatus. All of this green thing is the Golgi apparatus. So either it goes over here to be used, or it can go to the lysosome to be um, degraded. That's one. So that's for things coming from the outside to the inside of the cell. Okay. And then for the things that are already there within the rough endoplasmic reticulum, so we have some proteins here to be translocated to the Golgi apparatus. They may end up um, dividing from the Golgi apparatus, and that could go back to the Golgi apparatus. Cool. And then after like a whole process is within here, then it can subdivide as well, go to the outside as secretory vesicles. Okay, that's in order to be excreted outside of the cell. We also have these ribosomes on the Golgi apparatus can also go back as these divided little pieces, either again to the Golgi apparatus, or they can be like moved like this one to the rough endoplasmic reticulum. Okay, so the blue thing that has transferred whatever was outside the cell to the cell is like this blue thing is the clathrin. Okay, for the page color small dots over here, it's the COP1, and we said that the COP1 would take things step back, like retrograde. They take things from Golgi to Golgi, or from cis Golgi to endoplasmic reticulum. So it makes sense. So it's either from Golgi itself to Golgi itself, or like from Golgi to the endoplasmic reticulum. This should be like this. Yep. Mm-hmm. That's COP1. For COP2, with the brown dots over here, it was from the endoplasmic reticulum to the cis Golgi, and makes sense over here. So it's moving from the endoplasmic reticulum to the Golgi apparatus. Um, yeah, and that's what the colors mean of the arrows. And that should be it, right?